Hello, welcome to Biology, the scientific study of life. Translation is the RNA-directed synthesis of a polypeptide. A closer look. We will now examine in a greater detail how genetic information follows from mRNA to protein, the process of translation. As we did for transcription, we will concentrate on the basic steps of translation that occur in both prokaryotes and eukaryotes while pointing out key differences. Molecular components of translation. In the process of translation, a cell interprets a genetic message and builds a polypeptide accordingly. The message is a series of codons along an mRNA molecule, and the interpreter is called transfer RNA or tRNA. The function of tRNA is to transfer amino acids from the cytoplasmic pool of amino acids to a ribosome. A cell keeps its cytoplasm stocked with all 20 amino acids either by synthesizing them from other compounds or by taking them up from the surrounding solution. The ribosome adds each amino acid brought to it by tRNA to the growing ends of a polypeptide chain. Molecules of tRNA are not all identical. The key of translating a genetic message into a specific amino acid sequence is that each type of tRNA molecule translates a particular mRNA codon into a particular amino acid. As a tRNA molecule arrives at a ribosome, it pairs a specific amino acid at one end. At the other end of the tRNA is a nucleotide triplet called an anticodon, which bears pair with a complementary codon on mRNA. For example, consider the mRNA codon UUU, which is translated as the amino acid phenylalanine. The tRNA that base pair with this codon by hydrogen bonding has AAA as its anticodon and carries phenylalanine at its other end. Notice or see the middle tRNA in this figure. As an mRNA molecule is moved through a ribosome, phenylalanine will be added to the polypeptide chain whenever the codon UU is presented for translation. Codon by codon, the genetic message is translated as tRNAs deposit amino acids in the order prescribed, and the ribosome joins the amino acids into a chain. The tRNA molecule is a translator because it can read a nucleic acid word, the mRNA codon, and interpret it as a protein word, the amino acid. Translation is simple in principle but complex in its biochemistry and mechanics, especially in the eukaryotic cells. In this kitchen translation, we will concentrate on the slightly less complicated version of the process that occurs in prokaryotes. Let's first look at some of the major players in this cellular drama, then see how they act together to make a polypeptide. The structure and function of transfer RNA. Note before that, back to translation, the basic concept as a molecule of mRNA is moved through a ribosome. Codon are translated into amino acid one by one. The interpreters are tRNA molecules, each type with a specific anticodon at one end and a corresponding amino acid at the other end. tRNA adds its amino acid cargo to a growing polypeptide chain when the anticodon binds to a complementary codon on the mRNA. The figures that follow show some of the details of translation in the prokaryotic cells. The structure and function of transfer RNA. Like mRNA and other types of cellular RNA, transfer RNA molecules are transcribed from DNA templates. In a eukaryotic cell, tRNA, like mRNA, is made in the nucleus and must travel from the nucleus to the cytoplasm where translation occurs. In both the prokaryotic and eukaryotic cell, each tRNA molecule is used repeatedly picking up its designated amino acid in the cytosol, depositing this cargo at the ribosome, and then leaving the ribosome to pick up another amino acid. As illustrated in this figure, notice that this one A represents, notice that, and the book. A tRNA molecule consists of a single RNA. A single RNA, notice that. Amino acid attachment site, this one, boom. Consists a single RNA strand that is only about 80 nucleotides long, compared to hundreds of nucleotides for most mRNA molecules. Because of the presence of complementary stretch of bases that can hydrogen bond to each other, this single strand can fold back upon itself, forming a molecule with a three-dimensional structure, flattened into one plane. To reveal this base pairing, at a tRNA molecule looks like a clover leaf. The tRNA actually twists and folds into a compact three-dimensional structure that is roughly L-shaped. This picture, notice that. The loop protruding from one end of the L includes the anticodon, the special base triplet that binds to a specific mRNA codon. From the other end of the L-shaped tRNA molecule protrudes its three end, which is the attachment site for an amino acid. Thus, the structure of a tRNA molecule fits its function. The accurate translation of a genetic message requires two recognition steps. First, there must be a correct match between a tRNA and amino acid. A tRNA that binds to an mRNA codon specifying a particular amino acid must carry only that amino acid to the ribosome. Each amino acid is joined to the correct tRNA by a specific enzyme called an amino acid tRNA synthesis. The active site of each type of amino acid tRNA synthesis fits only a specific combination of amino acid and tRNA. There are 20 different syntheses, one for each the amino acid. Each synthesis is able to bind all the different tRNAs that code for its specific amino acid. The synthesis catalyzes the covalent attachment for the amino acid to its tRNA in a process driven by the hydrolysis of ATP. The resulting amino acid tRNA, also called an activated amino acid, is released from the enzyme and delivers its amino acid to a growing polypeptide chain on a ribosome. The second recognition step involves a correct match between the tRNA anticodon and an mRNA codon. If one tRNA variety existed for each of the mRNA codons that specifies an amino acid, there would be 61 tRNAs. In fact, there are only about 45, signifying that some tRNAs must be able to bind more than one codon. Such versatility is possible because the rules of the base pairing between the third base of a codon and the corresponding base of a tRNA anticodon are not as strict as those for DNA and mRNA codons. For example, the base U at the 5 end of a tRNA anticodon can pair with either A or G in the third position at the 3 end, which means of an mRNA codon. This relaxation of the base pairing rules is called wobble. Wobble explains why the synonyms codons for a given amino acid can differ in their third base, but usually not in their other bases. Okay, thank you.